In recent years, the price of fossil fuels, especially oil, has increased. At the same time, it is a political objective to reduce CO2 emissions from our consumption of energy. There is a big demand for development of a new generation of sustainable biofuels that don't require food to produce energy in order to meet transport needs in a sustainable way. In order to gain more knowledge, the Danish Council for Strategic Research has granted €3 million for a new research centre, Bio4Bio, at the Bioscientific Faculty of the University of Copenhagen. The centre works to develop new biotechnical methods that will make it possible in the future to extract biofuel from parts of cereal plants that are not used for human consumption today.
Så vi har brug for en ny teknologi. Det er en teknologi, hvor vi kan bruge det biomasse plantekulstof, som der er milliarder tontage i form af stængler og strå og blade og avne og alt muligt andet godt.
Today, straw and plant stems are considered waste products, but they contain a good deal of sugar that can be fermented into bioethanol. The centre is in the process of improving the technology for extracting sugar from straw and plant stems. How is this feasible? First of all, it is important to understand the molecular structure of plant cell walls. What chemical structures make some plants more suitable for biofuel production than others? Secondly, the centre is discovering new enzymes that effectively break down straw and plant stems so that they can be fermented into bioethanol. Several organisms, particularly certain fungi, are being examined, as they usually contain more enzymes which may turn out to be beneficial in this context. It is much more difficult to decompose straw and plant stems than leaves. The process takes more time and requires more energy as well as more enzymes. As we continue to acquire more knowledge, a solution is coming within reach. Another solution is to let plants themselves demonstrate their capabilities. This field appears to contain similar wheat plants, but 3 individual plants may be much easier to transform into biofuel than the rest. For example, because they require only half as much enzymatic activity to decompose. Researchers will focus especially on those plants. What makes the 3 plants unique? Why are they slightly different from the rest? Researchers can only identify those 3 plants if they examine all of them. Therefore, the centre has installed a robot-operated system that allows researchers to grind plants, weigh them, and analyse the characteristics of the plants by adding different liquids. The robot performs 500 analyses per day, while researchers were formerly able to carry out only 5 analyses daily. This will allow them to locate the needle in the haystack wherever it may be. Thus far, it has often been the case that the simplest characteristics decide the quality of the plants. One plant may have far fewer leaves than others, and since leaves are easily decomposed, researchers are happy to find specimens like this one. The goal is to locate and cultivate crops that are both optimal for food production and in terms of biomass. Therefore, it is not a question of producing food or fuel, but rather a question of being able to produce both at the same time. The use of biomass for energy and transport purposes is expected to increase considerably in connection with the transition towards a fossil-free energy system. Bio4Bio can contribute to making the production of second-generation biofuel more effective for companies and for the development of new efficient production facilities.